Hi, my name is Tonka and I have prepared a presentation about the sarcoplasmic reticulum. So, sarcoplasmic reticulum is a special form of endoplasmatic reticulum, and it is a membrane organelle in the sarcomere, wrapping contractile myofilaments inside the striated muscle. It is specialized in the lipid synthesis, calcium modulation, and excitation contraction coupling. Junctional sarcoplasmic reticulum is in close opposition to transverse tubules, which is essential for excitation and contraction. After an action potential, calcium is generated at the interface between transverse tubules and junctional sarcoplasmic reticulum and increase calcium concentration inside the cell, which causes the contraction of muscle. Sarcoplasmic reticulum serves as the largest calcium store. The endoplasmic reticulum calcium ATPase is responsible for active pumping calcium into the reticulum. The lumen of the reticulum contains calcium-binding proteins such as calreticulin and calsequestrin. Calcium release channels are responsible for the controlled release of calcium. The two classical calcium release channels are IP3Rs and the ryanodine receptors. The ion permeability of the IP3Rs is regulated by inositol, IP3, cytosolic calcium, and ATP. When a stimulus is received, IP3 is produced in response to the presence of IP3. The IP3Rs release calcium from the sarcoplasmic reticulum. Ryanodine receptors are activated by either calcium influx or after interaction with the dihydropyridine receptor of the T system membrane during depolarization. And sarcoplasmic reticulum can be divided into two distinct domains we can see on the picture. And is the longitudinal, which is formed by interconnected tubules surrounding myofibrils, and junctional, which is formed by the terminal cistern of the longitudinal reticulum set side by side with T tubules. Releasing calcium from sarcoplasmic reticulum to sarcoplasm allows for myosin heads to slide on actin, and muscle is active and in contraction. That's the purpose of calcium in our muscles mostly. But muscles also need to relax, which prevents them from muscle damage. And when calcium is not in sarcoplasm, then the muscles can relax because there is no contraction caused by the calcium. So free calcium can go back to sarcoplasmic reticulum. And two transporters to transport proteins on sarcoplasmic reticulum membrane are used for transporting calcium back to the reticulum using ATP, which means that this is active transport. So generally speaking, sarcoplasmic reticulum, as I have already mentioned, is the biggest calcium storage, and proteins such as calsequestrin, they concentrate calcium inside the cell and prevent it from leaving. So concentration of calcium is high inside, so that's why we need ATP as active transport to pull calcium against its concentration gradient. So first way how it can work is using the ATP calc is using ATP, and the calcium ATPase pump, and it moves calcium inside from sarcoplasm and releases hydrogen protons outside the reticulum. And the second way how we can do it, using the secondary active transport, is the sodium calcium exchanger, and this is generally the flow of sodium and calcium ions, where sodium is low inside the cell and in a little bit higher concentration in the sarcoplasmic reticulum, so it can exit through channels from reticulum to sarcoplasm without energy needed, and this moves calcium inside using also active transport. So these two processes help with calcium moving back into reticulum, which prevent calcium then from binding to troponin, which is bound to this substance, changes the shape of tropomyosin, and this led myosin attached to actin, causing contraction of muscles. But this prevention makes sure that muscles can relax. And also calcium can move outside the sarcoplasm and cell through protein channels in membrane using also secondary active transport and ATP. Thank you for watching, and these are my sources.